Dr. Magliocco to the podium, and he'll present cancer diagnostics and GIs for us.
Thank you, everybody. It's a pleasure to be here. I want to thank the organizers for inviting me. It's my privilege to be here to speak. I want to thank Sarah Loudon for a great conference, putting it all together. Wonderful format. Great to have the small setting, great venue, and being able to interact with everybody here to learn from each other. So these are very much learning events and very interesting times in cancer pathology. For those that don't know me, I'm a pathologist. I've worked in cancer for a very long time, for over 30 years. I was trained in Canada, worked in a variety of practice settings from the Fox Chase in northern Saskatchewan. I worked with the cancer program there, and in Alberta for about 13 years. So had the privilege to work on the introduction of HER2 testing into Canada and EGFR and other tests. Came to the US about 12 years ago to work at Moffitt Cancer Center, where I had the opportunity to lead the department there through the times of introduction of precision medicine and so on. I left academics about 4 years ago to work in private industry, really with a focus on underserved populations and trying to bring cancer diagnostics more globally, more nationally, so that more patients can get access to care. So today I'm going to be talking about advances. Those are my disclosures that you can look at. We're going to talk about advances in companion diagnostics with really a focus on GI. But one of the things that's happening is that a lot of the diagnostics are actually becoming pan-tumor indications. So in the old days, we really focused on treating breast cancer and colon cancer and lung cancer, but we're seeing more now where cancers have similar biology across sites, and we can learn from other cancers as well. So advances in breast cancer might now have impact on colon cancer, and we're seeing that, for example, with HER2 and so on. So I'll talk about some of the targeted therapies, some of the new platforms that are coming. We heard some great news yesterday. So I won't duplicate that, but really make some comments about that, and also talk about genetics and some of the future technologies that are really going to impact the way we diagnose and manage cancer. Now, as you all know, we're living in the revolution of precision medicine, where we're moving from one size fits all. And again, I've been lucky to live through this. Back in the day, when I trained, there's really only radiation and surgery and maybe some endocrine therapy, which they gave to everybody with breast cancer, regardless of their ER status. So times have certainly changed, and Sarah talked about rare diseases, but in fact, every cancer is becoming a rare disease now when we really individualize treatment for each person. We also heard about rare cancers becoming chronic, and hopefully that will be the case for people with common cancers like breast and colon and other cancers, that we're able to manage them over the long period of time. And also that these patients are being able to be managed at home so that they can really, their lives aren't disrupted, and they don't have to travel large distances to go to academic centers and others, that they can really be treated where they live, and that'd be really the vision. Now, if you're feeling overwhelmed, everyone is, and I just put this slide up to show you the impact. So on the left here, this is the number of FDA approvals per year. And back when I was learning pathology, we got maybe one a year, some none a year for cancer. Whereas I was looking last year, there's something like over 240 approvals for cancer. I'm on the alert system. I get 2 or 3 a week, it seems. And about a quarter of them are tied to companion diagnostics. So this is, this is riches. This is really a time, a renaissance in terms of opportunity for treatment, but it's also a huge challenge. How on earth can we manage this? This central chart is showing the potential companion biomarkers that are going along with these drugs, and about how are these going to ever get introduced into hospital care. And then over here on the right is an indication of the available drugs for each cancer. Each cancer is a red dot. And even in an academic center, that if someone's focused on one cancer, it's overwhelming. I can't imagine how challenging it is for a generalist who has to deal with multiple cancers that are changing all the time. Now, these are just individual drugs. Now we understand that there's a lot of studies looking at triplicate, at combination therapies, at phasing. How on earth are we going to manage that? Because if you do the math, you're looking at 68 million combinations of therapeutics, and how can studies possibly be done if we continue to do phase 2, 3 trials? So this is both an exciting time, but it's a challenging time. It's something we really need to think about. Now, the friendly neighborhood pathologist, I was looking for a picture of a pathologist last night, and I found a colleague here, this Dr. Wong, who practices in North Carolina. I don't think I know him personally, but the reason this photo appealed to me was this object here, which is an important tool that a pathologist will use, and certainly attracted me to pathology. That's a coffee cup or a teacup. And pathology was one of the disciplines where you could actually visit your patients and also enjoy a nice morning coffee as well. So that's one of the advantages. But pathology is really central because when you really think about it, no one has cancer until a pathologist says they have cancer. And so getting to know your pathologist is really important. And I think pathologists, a lot of them are focused now on trying to make the diagnosis, but I think that in pathology there needs to be a change about integrating pathologists more with oncology practices and informing pathologists more about treatment and the role of companion and predictive biomarkers, which pathologists are really in charge of delivering. Now, I put this slide up. I've used this slide for basically decades now, and it keeps coming back. This is HER2. And HER2 was through my whole career. When I was a resident, it was first discovered. Dennis Lehman published the first papers on it, and then continued to run the HER trials and so on. I was lucky to be involved to enroll patients onto those studies and actually bring HER2 in as a routine diagnostic in Canada. And there's many, many challenges to that. It took actually years and years and years to be able to get that test approved by the government and get it in utilization for all women. So it is a real challenge. A couple of things about it. It's quite an interesting target in that in the 3+, and this still kind of, I'm curious about this, that in cancers like breast and others, that there's a subset, about 15%, that have massive overexpression of HER2, and the gene copy number goes up over 100-fold. I don't know why that happens. I don't know if anyone does, but why do some people get that? But anyway, this is a very attractive target. And the nice thing about it, it's also coupled when you look at the gene copy number. So typically you score it from 0 to 3, and then if it's 2, you resolve it with using a FISH or a DISH method. Now this is great. We've been doing this for about 30 years now, but most recently in breast, there are new drug therapies, conjugates, that are focused and show to have efficacy in the 1+ and the 2+ that are not amplified. And I expect these will also make an appearance in colon and other cancers soon. And this is causing a challenge for pathologists because right now it didn't matter if something was called 0 or 1, so pathologists don't really care about that and don't look at it carefully. But if it became important, it's really imperative that the pathologist learns how to measure these things and identify the 0s and 1s. So that's a new challenge that's coming. Another thing that's quite interesting is when you look at HER2 in the gastric area, that I was involved in a lot of work trying to implement this in Canada again, and it can be quite heterogeneous. Here's an example of a gastric cancer where one half of it is highly expressed, the other half is completely negative. So you can imagine if you took a biopsy here, you would get a negative result, and if you did a biopsy there, you get a completely positive result. So this is just to remember or let you know that tumors don't read the books. They do what they want, and they can be heterogeneous. And this can be a challenge when selecting tissue for doing tumor-informed assays and other things. So it's just something to be aware about, that if you do get a negative biopsy, you may want to repeat that if you have broader tissue or so on, especially if you want to try to give the patient a chance at additional therapeutic options. Now, another thing that impacted pathology was the pan-tumor indications, and there's a great one here with mismatch repair. And pathologists can actually see mismatch deficiency with immunohistochemistry. So there's a number of genes called MMR genes, which are the mismatch repair proteins, and they get deleted or suppressed in cases where mismatch repair MSI high occurs. So it can actually be visualized in the path department without doing any molecular. Sometimes it'll reflex to MSI, but the power of IHC is you can apply this to a very tiny piece of tissue. So even if you have a few cells, you can actually determine if there's a deletion of those proteins. So it might be helpful if you're really struggling with the patient looking for options. Another IHC I'd just like to point to is how not to implement an IHC is PD-L1. And PD-L1 is causing significant challenges in the pathology industry in that many trials were done with PD-L1 as being the companion diagnostic, but they weren't really coordinated. So it ended up having multiple choices of clones, multiple cut points, and different ways to analyze them. So now we're faced with, when we're looking at PD-L1 assessment, labs have to consider what is the correct clone. They run on different instruments, and they're actually scored in different ways. So this is the scoring scheme. I won't go through all of this, but it's very, very complicated where pathologists have to assess both the cancer cells and the immune cells and come up with formulas that determine cut points and then assign, these are then used to assign patients to treatment. And it's quite perplexing given this is a single protein that's involved in cancers that we've devised a system that's this complicated. Now, going back to Canada, there was a period of time back in the early 2000s where it was discovered that one of the Canadian provinces, Newfoundland, happened to be running estrogen receptor not exactly correctly, and that they were underdiagnosing people with ER positivity, so that, in fact, it turned out they were doing this for 10 years, and every woman in Newfoundland was impacted by it, where basically many of them were called ER negative when they were actually ER positive, and the problem was the assay wasn't calibrated because the wrong controls were being used. Now, this isn't a problem of the lab, it's really a problem. This is, again, a thing. This isn't a problem of people. This is a problem of systemic problems where if we don't have proper tools in place or procedures where we can share and standardize assay, things will happen. Now, currently, there's a testing program where samples are sent across the country, but you can see even today where things are trying to be calibrated that if a sample goes to one lab, it may be negative, same sample, another lab may be positive, and so that there's problems with this can impact 5 to 10 years of therapy. So it's something that we really should try to figure out how to get it right. But the fact is it's complicated and it's hard because of the pre-analytics, the variability in testing, and the ability to analyze this testing. This is HER2 data here. You can see some labs struggle getting results. Again, these are from a few years ago, but it really points out to the problem of interlab variability. Now, new technologies that might make an impact, I'm quite interested in something called multiplex image analysis, and this is a piece of tissue. It's actually stained with 7 different markers in one cocktail, and it's really quite exciting. Not only does it produce a nice, pretty image compared to those brown images that we get tired looking at, but we can see now a nice image with all of these multiple light bright type images. But what this can do as well is it allows the computer to isolate the different channels. So it can take this image and pull it apart. So this is a cancer. This is actually a penile cancer. It was done at Moffitt. And this is the keratin channel. So you can see the keratin component. Cancer cells, these are CD8. You can see where those are, that those are in the stromal area. There might be 1 or 2 in the keratin area. You can see the FOXP3 that are basically exhausted T cells and where those exist. PD-L1 that's on the various lymphocytes, or PD-L1 that's on the various cells, PD-1. And some people, I like to point that out as well. We tend to use them interchangeably, PD-L1 and PD-1. They're actually 2 different things, and some drugs are against PD-1 and some against PD-L1, and their profiles are quite different as well, and this might explain a difference in outcome. And here's CD3. But by being able to do this, you can move it away from a human trying to score this to a computer that's actually measuring these exactly and counting them. So we might be able to standardize that, although it may give the illusion of accuracy. There's still issues with pre-analytics and the performance of these different antibodies across platforms, but it is moving towards more computational. And if that isn't enough for you, we're really just focusing here on PD-1, PD-L1, but there's a whole variety of other immune regulators that pharmaceutical companies are quite busy trying to make targets and therapeutics for. Another area I'm pretty excited about is something called image analysis with artificial intelligence. And this is where you can actually teach a convoluted neural network or other approaches pathology. And these systems can learn things that humans can't, because they can spend the equivalent of 1 million years studying slides and look at 100,000 of them, whereas your resident will obviously tire before you put them through that type of work. So here's an example where we taught a computer how to find Barrett's esophagus and actually predict whether or not we'll progress to become low-grade dysplasia. So this is an interesting tool where AI may actually see things on H&E that a human can't, and can even predict whether a tumor might have an NTRK mutation or an EGFR variation just by looking at the H&E. And this could be a low-cost way to quickly stratify a pathology and also deploy globally in low-resource environments in Africa, other parts of the world, where image analysis could really help with rapid diagnosis and selection of patients. And this is a technology could be very low cost and have very, very impactful technology. Now we'll need pathology and go into molecular pathology, and obviously somatic mutations are of high interest. We've talked a lot about them today and over the last few days, and there's plenty of different kinds of them, and I won't go into all of them. But really, if we put them into broad groups, the first group I like to think about are the canonical mutations and oncogenes. These are things like KRAS and EGFR and so on. The nice thing about those are extremely well characterized, and those can be measured super quickly. In fact, there's some assays that can do these measurements within 2 hours. Now, those could be really helpful, and they can work on really tiny amounts of tissue and can help get patients onto early treatment or onto trials quickly. So those are the canonical mutations, that there are special assays that are specifically designed for those. And those are somewhat limited, but we know that they have a high impact on a lot of diseases, including lung cancer and now colon and other cancers. What's really, a lot of people are excited are the broader panels, the general purpose panels, and there's many of them now in production. You know them of names like Foundation Medicine, et cetera. And there's ones from Illumina, TSO 500, et cetera, that have a broad global impact. These panels are generally the same, but when you want to look at them and compare them, what you really need to look for, are they DNA or are they DNA plus RNA? And that's one of the fundamental differences in them, not just the gene copy number, but do they have RNA in them? And the RNA can be quite valuable, because the RNA can tell you whether there's fusions there that may be missed by DNA. So you really want to think about how is the panel performing, what evidence is behind it. And here's the same panel can be used for all of these diseases. So that's why it's a general purpose panel. You can really use it. Also, a broader panel can tell you things like microsatellite instability, tumor mutational burden, and also homologous recombination deficiency. So these are very powerful panels, and they can detect fusions very easily within the DNA. And also, if you have RNA tracks, you can also see things like alternative splicing and different start points. And these can be quite interesting. There's some groups interested in looking at the RNA in terms of developing neoantigens and vaccine therapies and so on. So having a panel that looks at RNA could be quite helpful. Sometimes you'll run both and the RNA will fail and you'll only get the DNA, so that can be a problem when you're running these complex assays. But really try to learn more about the assays you're using and the power that's hidden there. I'd also say we throw away about 99% of the assays data because of the complexity of analysis and so on. Here's another technology that I found quite interesting. This is called optical genomic mapping. These are actually individual strands of DNA that are very long, hundreds of thousands of base pairs long, that are labeled with CRISPR at landmarks. And they run through these little channels and they're photographed, and the computer can actually assemble this, so it can create the chromosomes by assembling this Denovo. And the nice thing about this is the computer can then assemble a novel genome from this, and it can identify fusions and breakpoints and deletions and amplifications that are missed by a next generation sequencing. So it's very complementary. Technology can run quite quickly. It's seeing a lot of use in leukemia currently because of the fresh tissue. That's one of the limitations. You do need fresh extracted DNAs, which makes it hard. This is the same patient. So I was playing around with it, looking at, this is an ovarian cancer patient. This is her primary tumor, and this is the ascites. And you can kind of see some of the subclones are there, but there's definitely tumor evolution and so on there that's quite interesting to see. Another alteration is tumor mutational burden, and there's a lot of data emerging on this. We can see some tumors have high tumor mutational burdens like melanomas, and others are low. And if we draw a line across there, even in some tumors like colorectal cancer, you can see quite a range. And there's some that have very high and some that are quite low. So tumor mutational burden is interesting, and it generates neoantigens. There may be more work on this in the future as we determine developing vaccines and so on. I was really excited to see the work over the last few days on Collagiocarcinoma, because this really looks like the poster child now for GI malignancies, and it's very similar to lung cancer, where there are many, many attractive targets in there. And so this will create an interesting challenge for us to be able to detect these and properly stratify patients onto treatment opportunities. Now, the day in the life of a pathologist, here's the kind of samples I often get. But they're like, can you analyze this for 6,000 genes? It's like, well, it's a little bit hard. And one of the problems is that sometimes pathologists don't know, and they exhaust the sample looking at diagnosing the cancer. It's like, we already know it's cancer. We know it's lung cancer. We really need to take a sample that we can use for molecular analysis. And when we get samples like that, that's where we strongly advise liquid biopsy. That liquid biopsy can be very exciting, very powerful. I won't go over everything we talked about, but just thinking about a tube of blood, I always think, wow, this is amazing. Tube of blood tells us everything, the history of a patient, their ancestors, whether they came from Mongolia, about who your father might be, and about what sort of medications you have, and vaccines and viruses, and whether you got cancer cells. So it's really quite a very rich sample. It'll even tell you what you ate for breakfast, so it's a phenomenal thing. But the thing I want to point out is that the tube is important, because there are different kinds of tubes. The stability is important. Some cells will only last 24 hours, others can last 2 weeks, and so on. So you really need to know about how to collect a sample and how to transport it, so you can get the maximum information from it. Within the plasma side, there's tons of information, also in the buffy coat and so on. Wonderful technology for monitoring metastatic disease, because the tumor leaks all of these different things into the blood that we can analyze. We talked about yesterday circulating tumor DNA, phenomenal technology, tumor-informed, tumor non-informed, will go on to work, fantastic. There's other stuff in there as well coming out, proteins that might tell us about immune response. There might be, we can measure the white cells. Are they reacting to the tumor? What's going on there? There's exosomes, where they're like little letters, where cancers talk to each other, sending little packets of information, these exosomes across the body and so on. So really an exciting area of work in both the areas of screening, detection, monitoring, and also clonal evolution. I want to point this out that, when you use a tumor-informed assay, it's fantastic to look at elimination of the tumor, but also there may be new clones that arise as a tumor undergoes change. And tumors evolve over time, and new treatment opportunities may arise. And interestingly, some treatments may actually accelerate the development of mutations, like certain types of temozolomide can induce massive amounts of mutations in tumors and cause tumors to evolve. So very exciting window into real time, and can be much more sensitive and accurate than radiology in terms of following tumors along. So I'll jump over that. I just wanted to revive an old technology I like, that circulating tumor cells was around for a long time. There's actually about 1,000 papers on this, that when J&J developed this technology, and it's still FDA cleared. And so it's very powerful. What it does is you fish out the cells using EpCAM, and then you put them on a slide, visualize them, label them for keratin, nucleus, and also for white blood cell markers, and an open channel. And with this, you can count the cells. So here's some of the early data out of New England Journal of Medicine, that if colorectal cancers had more than 3, they did really poorly. If they had less than 3, they did better. So again, very powerful. But where I think in the future this comes in is we can visualize the proteins, particularly things like HER2 on the surface, and PD-L1s when we're considering antibody conjugate therapy, that I'd still keep this in mind as a technology. And this is going forward as well, that other companies are going to look at. This is a company that puts all of the cells on a slide. They can visualize the tumor cells and actually pluck them out and sequence them. They can look at the cell-free DNA separately, so you can do these very composite analysis and so on there. There's also other things in the blood, even from a drop of blood. You can measure things like proteomics, and some really cool things are happening, whereas you can extract the living cells and weigh them. Why the heck would you want to weigh a cell? Well, it turns out weighing cells is the most accurate thing in the world, that they sit on little atomic diving boards, and you can weigh them to extreme accuracy. And if you treat them with therapy ex vivo, they may gain or lose weight as an indication of sensitivity. So that means you don't have to culture them, and you can get a readout very, very quickly within an hour or two whether they're sensitive to a therapy. So keep your eye on some of these technologies that'll be coming along. I know I'm running out of time. If you bear me a minute or two, I'd just like quickly go over genetic testing. I think genetic testing is an area that will have more of an impact. You need broader panels. The cost has fallen dramatically. You can do whole exomes now for as low as $299, and that information is phenomenal. We can look at it for inherited mutations, but also polygenetic traits, other things, other comorbidities, et cetera. So I'd say genetics is going to make a big impact. And if we look through cancers, up to 20% of cancers show evidence of inherited single-cell alterations that could be related to the development of cancer. So I think we need to keep an eye on that as well. A few quick things towards the end. Tumor boards, phenomenal. They're easy to do at large centers where everyone's at the one place. In the community, it's harder, but we're very excited that we're working with some industry leaders to bring tumor boards to everybody. And there's some tumor board tools where this can be delivered virtually over time and space, where all of the information is in the tumor board. The case can be organized. There's tools there for trial matching. There's tools for evidence guidelines. And the individual oncologist can practice singly, or they can bring their colleagues in, or other experts can join them on these tumor boards, and the patient can be shared easily across the system. So virtual tumor boards, I think, are pretty exciting and something that we should look at. Here's some data showing that if a patient engages in a tumor board and that the actions or the recommendations are followed, they actually do better. And so this might be an argument for insurers to cover tumor boards and to help foster them as well. Now, one final point. This is an interesting study looking at data from over 50,000 Americans that were analyzed in claims. It was done to look at gaps in care, and they selected non-small cell cancer to look at the delivery of EGFR sequencing, and they made a model of what's happening nationally. And this is the predictive model of the state today. It's actually the state in 2019, because that's where the data was from, based on over 50,000. And they found that there were 7 gaps in the system as to whether patient had testing, whether testing was completed, even if it was ordered, because sometimes you'll order it and it couldn't be done, if the results were returned, and if it was acted on. And actually, out of 1,000, currently only about 356 patients are actually getting all the way to treatment. And again, this is national. This isn't one place. This is across the country. Obviously, some places are doing way better than others, and some are doing worse. I think this is a fundamental problem. And the uptake of any new technology takes time. For television and radio, internet, it took 14 years to get to 40% adoption in the general population. I think it's similar in precision medicine, so that's one of the great challenges. So I think we're living in a very exciting time of amazing technology. There are many barriers. How do we really get this out? And working together, we really need to work together and learn from each other. The systems, unfortunately, are fragmented. The data is siloed. The insurers don't really help us a lot, and there's payer problems and all kinds of things. But I think we all are in this together. Patients are at the center of this. If we keep patients front and center, we can really move forward and make a huge difference. So thank you for the opportunity to speak to you today. I also have a podcast that's designed for patients. It's non-commercial. If they want to learn about precision medicine, there's episodes that come out every couple of weeks. So thank you so much for the opportunity to speak today. I don't know if I have time for a question or not, but thank you.
One or two questions if anyone has any. Okay,
Thank you so much.
Thank you. Next up, I'm going to invite